All right, so I'm Laurie Barge at NASA JPL, and I'm going to be talking today about abiotic and prebiotic organic synthesis. And what I'm really interested in is how the conditions on planets, in particular in aqueous mineral systems, can affect the organic distributions that are formed from abiotic organic chemistry. And so there's a lot of different worlds and planets to be considering here when we're talking about the organic inventory of astrobiologically interesting places where we might want to send missions, such as Mars, the icy moons like Europa and Enceladus. But a lot of what we are talking about from the origin of life perspective, of course, is the early Earth, as shown here in an artist's conception. And so what I'm going to focus this talk on is when we have these planets or worlds with aqueous systems where organics that are coming in, say, from primordial sources can then be processed and further reacted in geological settings, either hydrothermal or otherwise, how can we look at the organic inventory of a sample or of a world and understand, first of all, where those came from, what conditions they may have experienced, and what organic distributions are likely to result from certain conditions, and then, of course, finally, how we can think about looking for life or understanding whether the origin of life is likely in an environment. So I'm going to focus mostly on early Earth because as a prebiotic chemist, this is a lot of what we think about, but early Earth was a very different planet than Earth today, and a lot of what we're talking about can be applied to ocean worlds or Mars or even terrestrial exoplanets. So early Earth, here's an artist conception. There's a lot of different artist conceptions when you look up early Earth, and this is because there's a lot of still debate about what these environments were actually like. There's some major outstanding questions, such as what exactly was the rock type that reacted with the ocean to give hydrothermal systems? What was the chemistry and pH of that ocean? What exactly was the atmospheric composition? How much land was there? And so on. But either way, it's a very big planet with a lot of different environments. And one important thing to note is that when we say environment, we mean a sort of type of place, like a hydrothermal vent or a volcanic island or something. But when we talk about reactions, we need to think about conditions, not environments, because conditions are specific things like the pH or the temperature or what the presence of an ion. And so in an environmental type, there's going to be a lot of different conditional spaces, and then there's also going to be a wide range of possible conditions for those environment types if we encounter them on other worlds. So for early Earth, there were a lot of different conditions that are good options for interesting organic chemistry. There were lots of different environments, including the ocean, the surface waters and deep waters, which would have given a nice pressure range. There was an atmosphere, and thus photochemistry and radiation could be prevalent. There's interfaces like the air-water interface and also the subsurface ocean interface. There's minerals and seafloor and, of course, hydrothermal systems. There was probably land, and so you also have on-land systems. And in these environments, we have conditions to consider for reactivity, so such as the pH of the ocean would have been strongly impacted by equilibration with a CO2-rich atmosphere, so probably around, say, 6 or 7. The ocean was anoxic, and so ferrous iron could remain dissolved in solution, which is a very reactive ion. But there was also Fe3 present because some photooxidation and oxidation from other ions could occur. There were metals, reactive minerals, and all sorts of environments where you can have low water activity, such as on land from drying, clays, gels, mineral surfaces, and so forth. And then, of course, pH would have varied quite a bit depending on where you were. So the ocean would have been around 6 or 7, but vents, hydrothermal systems, can vary in pH wildly from, say, very acidic, like a black smoker, around pH 2 or 3, to an alkaline vent, which can be up to, say, pH 11. So we have a lot of different environments, each containing multiple different reaction conditions. So as an example of an environment type that is containing many different conditions on Earth, I'm going to talk about hydrothermal systems because that's one thing that we specialize in our research group. But the same concept is applicable to other types of environments as well. So consider that when looking at a certain environment type, there were many permutations of conditions that could exist there on early Earth or on other worlds, and those could thus affect the organic chemistry that results in that system. So an example of an environment with many varying conditions is hydrothermal vents, which I know many of us are familiar with. But here is kind of the anatomy of one type of hydrothermal vent. Basically, it's a system where the seawater reacts with the ocean crust, and the seawater comes in through cracks in the crust and chemically reacts with that rock. And that fluid changes and becomes more, let's say, acidic or alkaline. It can contain dissolved ions. It can get heated. And then when that fluid comes back up into the ocean, it's a contrasting chemical fluid with the seawater now. And so it leads to the precipitation of minerals, either at a chimney, which is the vertical structure that forms at vents, or in the plume, which is like the black smoke from the black smokers. And these minerals in the plume can then kind of move out from the vent and settle out as sediments. So you also have hydrothermal sediments. And so in a vent system, even in a single vent, you have many microenvironments that are present there where reactions could be occurring. So you have, for example, the rock underneath the system where the water is reacting with that rock. And the type of rock is important, and the type of chemistry of that seawater is important, because that's what leads to the types of things that would be coming out in that hydrothermal fluid. There's the chimney itself, which is an interesting case because it is where the gradients are pretty strong between that hydrothermal fluid and seawater, and they can contain reactive minerals that could be catalysts for organic chemistry. There's the plume itself, which is full of mineral particles. Those are reactive too. And then hydrothermal sediments, which are pretty massive, and they have a large surface area. So these are also a reactive area where you could imagine organic chemistry. So even in one specific vent, you have a ton of different conditions that could exist, depending which exact microenvironment you're talking about. But hydrothermal vents on a planet are going to be widely varied as well. So as an example from modern Earth, there's a lot of different types of vents, and I just selected a few that I think are interesting and representative of the variety of conditions. And so vents on Earth range from very acidic, high temperature, high pressure black smokers that have a lot of reactive minerals like metals and metal sulfides. There's hydrothermal seeps that kind of bubble out, say, CO2 and other gases. These could be rich in, say, silica or heavy metals. There's lots of different alkaline vent types, like, for example, Lost City or Prony hydrothermal field or Streetan in Iceland. These can have more alkaline pH, but their mineralogy can vary, for example, from carbonate to silicate. And then vents can also occur at all different depths. So you have very deep vents, like kilometers deep, where then the pressure would result in very high temperatures, like several hundred degrees Celsius. But also vents can be shallow enough that a diver can reach them, that light can penetrate to them. And in one case, this case, the vent is actually exposed to air periodically as the tides go in and out because it's that shallow. So there's all these different types of conditions that can exist in hydrothermal systems, and these are just some on Earth today. So considering early Earth would have even more conditions, and then if we talk about other planets where there may have been hydrothermal chemistry, or even still today, those have still more sets of conditions. So one example is Saturn's moon Enceladus, which is interesting because it's been theorized that there might be hydrothermal systems there today. And if so, those would be water reacting with the chondrite core. The ocean is thought to be alkaline, and what exactly those events would be like is unknown. And so hydrothermal systems contain many different conditions. And so when trying to figure out what organic chemistry is likely to occur in these systems, we need to consider how specific conditions like this, like pH or redox state and so forth, would affect a certain organic reaction. And furthermore, for example, when talking about ocean worlds, these are interesting for astrobiology because they are places thought to be potentially habitable, where maybe origin of life could occur, or where maybe hydrothermal systems could exist either today or in the past. But ocean worlds are each different from one another as well. And so when we talk about, is there likely to be organic chemistry happening on Europa or Enceladus or Ceres or any of these other bodies, the conditions specific to that world and specific to the environments on that planet are going to determine what organic chemistry is favored. So they're not all the same, and things that would affect the organic chemistry on these bodies would include the rock type, the ocean chemistry, the pH, the temperature, the degree of differentiation, for example. Like Enceladus, because it's so small, it never melted, and so then you would have a lot of primordial organics present from that chondrite core. So considering specific conditions for any ocean world is really important when trying to understand what abiotic organic chemistry is favored. So that's a general story, but I'm going to give you one example of a reaction, a specific reaction that we study in the lab, just to show you how conditions can affect an organic chemistry reaction. And this is intended to look at a simple reaction that we understand pretty well as far as mechanisms and what products you expect, but the relative abundance of products is strongly affected by the environmental condition. This is one example that we study, but it's meant to be a general point that any organics that we're looking at that are abiotically produced or prebiotic, we should consider how conditions might affect those relative abundances as well. So this is a reaction that we've studied extensively. This is a reduction in reductive amination chemistry of carboxylic acids, pyruvate and glyoxylate. And these are interesting because these are found in life. They're important for biochemistry. They're also thought to be relevant to the origin of life. Specifically, they're studied a lot as kind of proto-metabolic reactions, making some products similar to, say, the TCA cycle, and thought to be maybe a precursor to some things that could have eventually become a biological metabolism. These types of chemistries and the products they make are also relevant for Enceladus because they're similar to some of the things that we think we've seen in the Enceladus plume. And so the reaction that I'm going to show, the way it works is we make a mineral precipitate of iron hydroxide by precipitating ferrous and ferric iron salt with hydroxide to a pH of 10 or 9, so alkaline pH mostly. And then we add in these organics, pyruvate or glyoxylate, and then we add a nitrogen source, in this case ammonium chloride. And this is representing a particular hydrothermal type of sediment or plume that could be coming out of an alkaline vent in an iron-rich ocean on the early Earth. But in general, you could permutate this kind of reaction to contain other organics or other minerals, different pHs, nitrogen sources, and so forth. But we're keeping everything constant and just varying the geochemical condition in order to show you how the distributions of the organics shown here will change. So we know what the products of this reaction are. It's expected that you're going to form the reduction products, the alpha hydroxy acids, lactate, and glycolate. It's also expected you're going to form amino acids from reductive amination. But when we change things like the iron redox ratio in these minerals, the ferrous to ferric iron ratio in the hydroxide, or the pH, or which nitrogen species we use or its concentration, the temperature, and also the presence of other things like phosphate, we do see differences in how these reactions turn out. So first I'm going to show some impacts of if we make this mineral and we change the redox state of iron in the mineral from reduced to oxidized, how we then change the reaction products that form and their relative abundances. So for a pyruvate reaction, again, the most common products we get are the amino acid alanine and the hydroxy acid lactate, so reduction and reductive amination. And in this chart, we have pH 10 and pH 7, and then there's the x-axis is the gradient of iron redox state. It's the fraction of iron 2 in the iron mineral that contains Fe2 and Fe3. So over here we have the completely oxidized ferric hydroxide, and over here we have the completely reduced ferrous hydroxide. And so what we can see is that the amount of alanine and lactate and pyruvate remaining after 3 days in this case, it varies depending on both the pH and the iron redox state at which the experiment was conducted. And in this case, the formation of alanine is only really favored at alkaline pH and at an intermediate iron redox state. So this is about the iron redox ratio where you would have, say, magnetite existing. And so we think if you had magnetite and some amorphous iron oxides and your alkaline pH, then you might make alanine. But in this reaction, if you were more reduced, for example, you would just make lactate. We see the same thing with glyoxylate. It's the same kind of reaction, just a different precursor. And again, we'll make the reduced product glycolate and the amino acid glycine. But here we see that, again, you do get different relative abundances of amino acid and hydroxy acid as a function of iron redox state and these two pHs. But it's also not quite the same as the previous slide, so you do get variation here, but the conditions under which amino acid formation is favored in this reaction is slightly different than the previous one. And so this indicates that probably when you have any specific carboxylic acid that's going to undergo this chemistry, each one is going to have a slightly different version of reaction product distributions that's going to occur at these different iron redox states. We also have looked more deeply into the effective pH, and so by keeping constant an iron redox state of 66% Fe2, which is basically where magnetite would form, and then at a temperature of 70 degrees Celsius, we see at different pHs for the pyruvate reaction, from 7 to 11, that we start to see an increase in alanine formation as we get more alkaline, but specifically between pH 9 and 10. And so the pH is really a big factor here in how this reaction can proceed, obviously. And so we think this is to do with the pKa of ammonia around 9.25, and that's maybe what's affecting how you will form the amino acid versus just reducing to form lactate. And so maybe if you have other nitrogen sources in an experiment like this, maybe the pKa of that is going to be a big factor in where the pH dependence lies. That's not something we've tested, but I think that would be an interesting future direction. And so when we're thinking about specific environments or specific, say, hydrothermal vents, looking at the environments I showed previously, there's these alkaline vents that range in pH from, say, 8 up to 11, like Lost City and Streetan and Prony and Old City. And so the exact pH of those vents, if you had all these other conditions, you might get a range of product distributions of, say, alanine versus lactate in that system. And then the Enceladus ocean is thought to be around pH 9 to 10. And so that would put you right here where really we see this escalation of alanine forming. And so this is why when we have missions, it's really interesting and important to know these geochemical conditions as well as detecting all the organics, because it really matters whether the pH is 9.5 or 9.75 or 10 in order to predict what the abiotic abundance of alanine should be in an Enceladus condition here. We've also tested the effect of minerals versus no minerals, as well as temperature. And as just some more examples here, the pyruvate reaction, if you do it at different temperatures, at a state of Fe2 at 50% and a pH 9.2, and we did this after 3 days, we see that the reaction, it basically speeds up with temperature up to about 50 C. And it's kind of the same here, more or less. But if you do it at room temperature, it's much slower. However, you can wait long enough and eventually you'll still start to make amino acids sometimes in these reactions. So this is a glyoxylate reaction that is run for 21 days. And you do see glycine forming, it's just much slower. So these have to do with the time scale, which is another important factor. So if you have an organic or prebiotic reaction that, say, is faster because it's catalyzed by minerals or because it's heated, then the kinetics of that might be affecting what happens downstream. So if you have other likely reactions, like, say, formation of polymers from your amino acids, then the kinetics of the formation of those monomers might affect what you get as you keep processing this toward a more complex prebiotic system. And then again, what environments might drive certain permutations of this. So here's several alkaline vent temperatures plotted over these results. And then the Enceladus core, I don't know what exactly that temperature is, but if it were, say, 80 to 100 degrees, you would expect to kind of get this ratio as opposed to something like this, which might be more prevalent near the seafloor. So when looking at ocean world samples, or say in a plume, where exactly did those samples come from and what temperature were they at when those organics were synthesized? That's an important question, because if any of this happens, say, in a sediment on the seafloor where it was colder versus in the core where it was more heated, you might get different distributions depending on the time scale of that migrating up to the plume. And then finally, another thing that we tested is the effect of the concentration of ammonia. And so in this case, the ammonia is what's, of course, giving you that amine group for making amino acids. And we did some experiments with glyoxylate at 2 different iron redox states. So 33% iron 2 and 50% iron 2, pH 10 at 3 days. And we increased the ammonia concentration from 0 up to 400 millimolar. And so we see in this reaction that the glycine formed basically increases almost linearly with the ammonia that we add, which kind of makes sense because you need the ammonia to make the amino acid. But what's interesting is the glycolate, the reduced product, seems to really more depend on the iron redox state, not so much on the ammonia concentration. So this is an important point too, because in a reaction where you have multiple organic products from the same precursor, you could have different conditions impacting those distributions, but each specific product, it's possible that they could be impacted differently by different conditions. So like you might have one product that's mainly controlled by how much Fe2 is present, but there might be another product that's mainly controlled by how much ammonia is present, and something else might be more controlled by what the pH is and so forth. So that's why this can get really complicated, and it's important to know the specifics of your conditions and how each product might be affected by those. And then again, here's with no mineral after 3 days, and we're going up to 200 millimolar ammonia. And after 21 days, we see the same formation of glycine. And again, it's linear with the ammonia concentration. It just takes longer. So when thinking about environments here, Earth vents are generally not going to have these huge amounts of ammonia. So you might be down here, and we do form amino acids in these experiments at low concentrations of ammonia, like you can get down to several millimolar only of amino acid. But then Enceladus, there's been some studies proposing that Enceladus might have very high ammonia concentrations. And so, would something like this or even higher off the scale that we tested be possible for Enceladus? Maybe. So we have done a lot of experiments in closed aqueous systems, trying to remove the effects of things like radicals and radiation. But of course, if you have a terrestrial body that's got aqueous systems, there's going to be atmospheric photochemistry and radical production and production of reactive species. And so here are some other species to be considering when we think about organic inventory on, say, the early Earth. But also, this is relevant to Mars, and it could be for any terrestrial planet where you've got an atmosphere. So if you have photochemistry, you can produce organic and different types of, say, nitrogen radical species and other reactive species. But in addition, you don't have to have an atmosphere or radiation to be doing this. It is possible to make radicals underwater with minerals and mineral reactions. So here are some examples of things that maybe we could consider for early Earth or ocean worlds, like production of hydrogen peroxide and Fenton chemistry underwater, or production of thiol or disulfide radicals through mineral chemistry underwater. And so any of these radical species, those will definitely affect these reactions. They could cause oxidation of iron. They could cause reactions of the organics. And that's something that would all have to be very rigorously tested in order to understand how production of radicals and the other species here could be affecting some organic chemistry on planets, even if those radicals are no longer present when you detect the sample. So another aspect is if you have organic chemistry that's being impacted by the environment, there's other parameters. It's a very complicated system. You've got all different ions and salts and so on. And so even if you have an environmental parameter that's not directly driving an organic chemistry reaction, it can still affect that reaction if it drives another parameter that does impact that reaction. So for example, I showed that the redox state of iron, the ratio of Fe2 to Fe3 is really important in driving the product distribution from, say, pyruvate and glyoxylate. So therefore, anything that impacts the iron redox state is going to impact these reactions. And so we've done some work on just looking at how other elements cycle and affect iron redox in early Earth systems. And for example, if there is nitrate or nitrite present, which there could be if you have photochemistry of a nitrogen-containing atmosphere, if you have nitrate or nitrite present with dissolved ferrous iron, then we observe the oxidation of iron over about 7 days. And this is true for nitrate, but it's a little slower, around 7 days. And it's true for nitrite almost instantly. So nitrite is super reactive with iron. However, we see that this really only occurs very quickly at pH 8 and 9.5, not so much at pH 6. So this one would be more favored in an alkaline condition. But then you might be changing the iron redox state more toward Fe3 if the pH was higher. We've also looked at mechanisms for reducing iron, which again would affect these organic reactions. So sulfide can be a great reductant for Fe3, and also sulfur-bearing organics, like for example, thiols, can be a good reductant for Fe3. So we've done some work looking at the amino acid cysteine, which is a thiol-containing amino acid. And over the course of 7 days in the presence of cysteine, we see iron reduction. So you can change the Fe3 back into Fe2. With no cysteine, we do not see any reduction of iron. So here you've got kind of a possible oscillating iron redox cycle, depending on the kinetics, the pH, the concentrations, and so forth. But even other things that are not, again, not specifically reacting here can affect the reaction. So we found, for example, that if phosphate is present in the nitrate reduction reaction or oxidation reaction of iron 2 hydroxides, the phosphate will bind to the surface of the iron mineral and prevent its oxidation, even if nitrate is present. So, again, like if you have silica in the oceans of early Earth, or phosphorus, or nickel, or any other of these other things, how is it going to impact the reduction of iron or the pH? And then those, in turn, could greatly impact the distribution of organics that could be formed. So putting some of the data I just showed together, we can make a plot of these organic distribution abundances as a function of geochemical condition. So here, looking at pH and iron redox state, of course, there's other conditions, but just simplifying it to this. And so you can see that it just really depends on what those conditions were in that organic system, what distribution you might be able to detect in a sample. So when saying, what is the abiotic expected distribution of amino acids in an alkaline vent, well, there isn't one answer because it really depends on those conditions and on all the specifics that might be affecting that environment. So, looking at this in an environmental context, say you started out with an anoxic ocean, so maybe some Fe3 and mostly Fe2, around pH 7 because it's in equilibrium with CO2, and maybe you've got some alkaline vent mixing. Maybe these are the products that you might expect to form. But if you have instead the same reaction occurring but in more shallow waters, so you have more oxidized iron because, let's say, the photochemistry is producing nitrite, which instantly oxidizes all that Fe2, then maybe you would get this kind of distribution. Or maybe if you were at a deeper system, just slightly deeper, where the radiation can't penetrate, where those oxidized nitrates and nitrites have already been consumed, so the iron gets reduced in the presence of, say, sulfide or thiols, and now it's completely reduced mineral, and it's more alkaline because it's near a vent plume, you might get this distribution. Or if you were up on the surface, let's say you were completely exposed on the surface, so all the iron just is getting oxidized by photochemistry, but you've got an alkaline vent like Prony, and it's making a very high pH solution. So all these are different examples of what you might find in different permutations of the same reaction, even in the same type of environment, in an alkaline vent, but just kind of within the microenvironments where that might exist on a planet. So this is something to consider for, say, Enceladus, Europa, Mars, and other places where we talk about organic detection, because it's not just what you have found in the sample, it's understanding where that may have come from, and thus, could this be a prebiotic system? But also, we can talk about whether things might be a biosignature or not. So when talking about all these different worlds, we've got all the ocean worlds, which are each different from one another. You've got terrestrial planets that may have had water in the past. And so of course, we want to look for life, right? And we want to detect organics. But even if life is not present, and even if it never was, we would expect to see remnant abiotic and maybe prebiotic organic chemistry on some of these solar system bodies. And it might even be pretty interesting, actually, to find a system where we have prebiotic or abiotic chemistry that never became life at all, because we might be able to wonder where that threshold is for determining whether something is alive or not. And so then my final point I'll make is that the threshold for where, if you detect an organic, if you could say it was from life or not, the threshold for being able to say that's life versus abiotic, it's not going to be one specific answer because the conditions are going to determine what abiotic chemistry is possible. So therefore, that threshold for identifying a biosignature would change based on the organic chemical history of that planet. So just as an example here are a bunch of organic chemistries and products that have been made in the lab. These are origin of life experiments from the literature. And there's been so much work done in this field, so it's things like monomers, like an amino acid, or an ATP, or a peptide, or an iron sulfur cluster, or a protocell with things inside it. There's all sorts of lab work in this field. And so these are all things that presumably for early Earth anyway, we think existed at one point abiotically because that's how the origin of life happened. And so if you have a world, say an aqueous mineral system like ocean worlds or a terrestrial planet, and you have organic chemistry that's approaching an origin of life state, what would it look like to detect something like this, like a complex, let's say, iron sulfur cluster nested in a peptide? And how would you know if such a detection was biological or not, even if the detection itself was perfect? And so we would suggest that if you are a prebiotic world where there's either no or minimal biology, such as perhaps Enceladus today or something, then that threshold where you would have to be to identify whether there's life based from organic chemistry would be quite high, because really complex organic chemistry that's kind of approaching an origin of life state is still a possibility. And that would look to any of our mission instruments like a very complex thing that could be confusing. However, if you were on a world, say, such as Earth, where we have life, and there's a lot of life on this planet, and you detect something that looks like any of this, it probably is life, and because there's a lot of life here and it's mostly suppressed the abiotic background, at least with these very complex things like prebiotic chemical states. And so the detection aside, where to place this threshold for where we could consider an organic chemistry or an organic inventory as biology needs to be kind of changing depending on the world and depending on its geological history. And that's why it's super important with missions to get this geological and geochemical data as well, so that you can try to place this threshold somewhere to help identify the origin of any organics we might find. Okay, so just some takeaways. First of all, as I've shown, prebiotic or abiotic reactions of organics are strongly impacted by the experimental conditions, which of course means that organic chemistry on planets or in environments is going to be strongly impacted by the environmental conditions that are present. I was also asked to talk about what sort of future work I think would be interesting for each of these. And so I think in this regard, it would be really impactful if that we had more studies of how various conditions in the lab impact organic product distributions of prebiotic chemistry. So we've done one type of reaction that's interesting, but there's all sorts of others in the literature, and not a lot of them have been thoroughly explored in terms of what conditions might do to those distributions. So I think that would be a really interesting direction. We also see that environmental parameters and conditions impact one another, sometimes in complex ways, which then can impact the organic chemistry. So for example, if you have a lot of nitrate in the waters with iron 2, then that's going to affect the iron redox state, which then affects the organic distribution. But we have to consider this for all the myriad of different geological conditions that could be present. So I think more studies would be needed of how geochemical parameters, not the organics, but the geochemical parameters, how those affect one another. So this would be elemental cycling of, say, phosphorus and nitrogen and sulfur and iron and other metals, and how that all impacts one another, because that can change fundamental properties like pH, for example, that could then impact organic chemistry. We know that environment types, such as, say, hydrothermal vents, these contain many permutations of reaction conditions. And so it's not enough to just say, we think this reaction could occur in a vent. We have to say, no, specifically we meant at pH 9.5 with the presence of iron, with the presence of radiation in a shallow water, and so on and so on. So it's important to study planetary environments and think about early Earth to determine what sets of conditions different environments might contain. And in particular with planetary science, this is important because we find out a lot of surprising and awesome things with missions. Like we find environments that contain conditions that we never expected. And so if you have a reaction that you know is driven by something specific like presence of an ion or pH, you might find that condition in some surprising environment on another world that we didn't expect. So that's why it's really important not to limit, thinking of an organic inventory as produced by an environment type, but more about the condition space in which it happened. Ocean worlds, which are super interesting for astrobiology, these each contain different conditions. So they're not all the same, and we have to understand the conditions in each of them that could impact the organic and prebiotic chemistry. So I think one thing that would be really helpful is more ocean world studies to understand geochemical properties such as the seafloor and the core, like the depth of that interface, the chemistry of the ocean, the pH of the ocean, the geological history of the world, the rock type, whether it's differentiated, stuff like that is super important for understanding the conditions that could drive organic chemistry. And then finally, we talk a lot about looking for life, but in order to have life, you have to have an origin of life. And so something we need to think about for other worlds is, are they likely to have driven the origin of life? Habitability is not the same thing. And so if we want to look for life elsewhere, we have to think about how origin of life could be driven on other worlds. And it's possible that these chemistries that we think are necessary for life to emerge, maybe they are necessary and maybe they could occur elsewhere, but the mechanisms might be different, the environments that drive them might be different. Or it's possible that you might have different chemical pathways toward a lifelike state. So all of this is something we need to think about and not just think, is a world a place where life would live if you put it there? That's not the same as saying life could emerge there. And so I would hope that we can start to simulate known prebiotic chemistries for Earth, because there's a lot of work done in this area for Earth. We could simulate those reactions under other planetary conditions to see what the outcome might be. And that might help us identify planets where we're most likely to have an origin of life. So that is it, and I'm happy to take any questions. Thank you for your attention.